Alfred Hershey and Martha Chase create an experiment to see how the T2 bacteriophage reproduces. They use radioactive isotopes of phosphorus and sulfur to track the location of the molecules. Phosphorus was used for the DNA and sulfur was to track the protein. This is due to sulfur being in the protein but not the DNA, and vice versa for the phosphorus. They then grew multiple batches of the Escherichia coli, better known as E. coli. The first batch was grown in a medium of radioactive phosphorus, and the second batch was grown in a medium of radioactive sulfur. The other batches were grown in normal mediums, and then they took the first and second batches of the E. coli and injected them with the T2 phage. The other batches were injected with phosphorus and sulfur-labeled progeny phages. The E. coli and the T2 phages were given time to grow and then placed the cells into a blender to remove the phage coats from off the cell walls. They cultured the infected bacteria cells and separated the protein coats. The sulfur phages did not show any radioactivity in the progeny phages, showing that the protein portion does not enter the cell. The phosphorus, on the other hand, infected phages did have radioactive progeny phages, showing that DNA was the genetic material of the phages. This means that the phage reproduces by attaching itself to the cell wall of the bacteria cell. The phage then injects its DNA into the cell, then replicates and makes the cell synthesize phage proteins. The phage DNA then encapsulates itself in phage proteins. These are considered progeny phages and lyse the cell to escape.